Right. Good morning, everyone, and welcome to the second day of the Neural Engineering Research Symposium, hosted by the Institute for Neural Engineering at University of Miami. My name is Surud Rajguru. I'm an associate professor in biomedical engineering and otolaryngology at the University of Miami. And along with Dr. Dalton Dietrich, who is the scientific director of the Miami project to cure paralysis, we co-direct the Institute for Neural Engineering. On behalf of all of us and the organizers, Hannah, Federica, Raquel, and Shu, we want to welcome you all to the second day. We had a fantastic first day yesterday with two excellent sessions, and we're looking forward to having three other sessions today. The first session today from 9 to 11:30 will be on the brain-machine interfaces. The second one will be on the aging brain, followed by we'll have a poster presentation, including live presentations. We also have awards for the poster presentation, so please stay on to hear about those. We'll have $250 each for the poster awardees, and we have 5 different awards to hand out. With that, we'll get started with the first session. I'll turn over to the moderators, Hannah and Federica, who are graduate students at the University of Miami and have developed the program.
Hi, everyone, and welcome to the session on brain-machine interfaces. My name is Hannah, and Federica and I will be moderating the session. Briefly, I just want to remind everyone that the talks are 10 minutes, and if you have any questions, just leave them in the Q&A section, and we will compile those and ask those in a 20-minute panel discussion at the end. So with that, we have an outstanding lineup of speakers, and I will just read the title so we know what's coming up. And as I'm reading the title, if the speaker can share their screen, then this should run pretty smoothly. So first, we will be hearing about physics-based computational modeling and neurotechnology, predicting performance of interfaces in the nerve and the brain.
Great. Hello, everybody. Thank you for inviting me. I'm excited to talk about my own research using physics-based computational modeling and neural technology. I have 3 examples that I'm going to showcase today. First, though, I wanted to kind of point out the state-of-the-art interface modeling in the literature. So an example of neuroprosthesis modeling from Silvestro Macera's group, pain management from Scott Lemke, and movement disorder application from Cameron McIntyre's group. And so in both Lemke and Macera's group, they used computational modeling to predict the amount or the ability of these either intravesicular in the case of the prosthesis or epidural electrodes to stimulate target fibers in either a peripheral nerve or the spinal cord. And so their modeling has been very useful in predicting the performance of these electrodes, and then would again help clinical therapies. And then Cameron McIntyre, I'm highlighting 2 pieces of his work. So one using, again, stimulation prediction of fiber activation. And in this, it's rather new. Before they did a volume of tissue activated approach, which did not take into account the actual pathway of the nerve fibers. So in his new work, they're incorporating the fiber tractography and getting even more patient-specific results. And I also want to highlight some of their older work that incorporated recording performance. So predicting the recording performance of neuroelectrodes. These are DBS electrodes in this case. So there's many uses for computational modeling, and this is not, of course, everything that's out there, just a sample of what's going on in the literature. So before I highlight 3 of our own examples of modeling, I want to talk a little bit about the mechanisms used for the biophysical modeling. So normally you have a finite element model coupled with a biophysical model. So the finite element model comprises the volume conductor, which can be as intricate to the neural target as you want. Essentially, this would be normally done in finite element software such as COMSOL or ANSYS. You're solving the Laplace equation and solving for the electrostatic potential as a function of user-defined conductivity. And so this is where you can make your model as specific as you want to. Here I'm showing a peripheral nerve where we would not model the fibers in the volume conductor model, but we would model the endoneurium, the perineurium, which encases the fascicle, the endoneurium, which is the stuff in between, and then the electrode array. And so the boundary condition, given to Laplace's equation would either be a current or a voltage boundary condition. And then from that current or voltage inputs, you would basically calculate a three-dimensional voltage. And that voltage then can be passed into the biophysical model. Neuron is a wonderful tool that a lot of researchers use. And more specifically, almost everyone in the field uses the MRG axon model. So this was made by McIntyre, Richardson, Grill in 2002. And it is a double myelinated, or it's a myelinated double cable model. So you have resistive and capacitive elements for the core conductor, and then resistive and capacitive elements for the myelin. And more importantly, you have dynamic conductance-based models for the ion channels at the node of Ranvier. And this is where the Hodgkin-Huxley formalism is used and incorporated to model the action potential dynamics. And so for stimulation, we would couple the voltages predicted in COMSOL to placements of either axonal fibers or neurons themselves. You can make compartment models of neurons using this approach. And then for the other way, to predict recording, we would essentially go the other way. We would use neuron to generate currents that would then be boundary points or current sources in the finite element model and be able to predict recording performance of the electrode. So going on for my first example, this is work that I've performed with Dr. Bashrula, Dr. Gunduz, and Dr. Otto a little bit ago. And we were also working in conjunction with researchers at IMEC who fabricated a intravesicular electrode for us. And we were basically assessing the feasibility. And so my team was in charge of figuring out, well, we wanted to be able to figure out if we could deliver a population-based code for tactile feedback. That was the end goal for this sensory neuroprosthetic. And so by modeling, we really wanted to see if we could quantify the extent of fiber activation in terms of how many fibers were recruited or activated with increases in the pulse amplitude. And so neural tactile sensation is encoded by spike rate, which is very fiber specific, but it's also encoded by quantity, how many fibers are recruited or activated for a given pressure level. And so we knew that we couldn't, these electrodes, it was really going to make any fiber specific stimulation, but we could have specific population stimulation. So that's what we were going for. And here's an example of some of our results. So these two figures are cross sections of a heterogeneous mixture of 2,000 axonal fibers, and we used the rat tibial nerve as our model, both in the experiment and in this simulation. And so each color represents a range at which that axon was activated. And so the population distribution is given over here. And so what was interesting to me was the effect of our volume conductor. So the left side, I used an isotropic indunarium conductivity and no paranarium, no epinarium, no other volume conductor other than one isotropic, basically big volume conductor. And that gave a very different activation response for using a more realistic modeling finite element model, where I did use anisotropic indunarium conductivity that has been given in the literature, plus a paranarium and an epinarium, and then a volume, saline volume around it. So you can see here the difference, how essentially it was really the paranarium that made the most effect. It's very resistive, and so it bounded the voltage essentially. And you could see a lot of variability in the activation here, basically because the fibers are randomly distributed, therefore their nodes of Ranvier are kind of randomly distributed around the electrode. But because in this one, the voltages are a little bit more bounded by the paranarium, it averages, well, it allows it such that now you have a much better relationship with distance. So showing the same results again, here's the recruitment curve for the anisotropic and isotropic case. And I want to highlight that in these figures, I'm showing the threshold current as a function of radial distance away from the electrode. And at very far distances, you see the normal recruitment order. It takes more current to stimulate smaller diameter fibers. These colors are now representing the diameter of the fibers. But still at the really low, very near the electrode, you have almost the same variability as you did in the isotropic case. And I think for for tactile feedback, we really want high resolution. We will want to pack the electrodes in and therefore work within a couple, maybe 100 or less microns from the electrode, only recruit the ones here. So we're still going to have to worry about this type of random recruitment order, essentially. So that was a key takeaway. And moving on to another application, I'm working with Dr. Jack Judy and his students, and he has he's also a fabrication engineer, and they have a tissue engineered electronic nerve interface that now has a lot of design freedom. And the question that they are wanting to answer is that for their planar electrodes that are disc shaped, is there an optimum diameter for maximizing the amount of fibers recordable? So we're switching, it's no longer really looking at stimulation performance, but now recording performance. And this could be an application for a bidirectional interface again. And so this is, is there an optimum diameter? And then also, is there optimum spacing and stacking of these electrodes to be able to maximize the amount of signals that could be recorded? So I first want to show you simulation results that are just showing the voltage as a function of time at specific singular XYZ points away from a myelinated fiber. So the node of Ranvier is here, and you see the biggest spike near the node of Ranvier, of course, and then it decreases as you go out. So depending on the diameter of your electrode that would just be sitting near this fiber, you would essentially spatially average over all of these voltages. And the diameter of your electrode could impact the amplitude of the signal that you are able to record. So we did more simulation of this, and this is again using the anisotropic volume conductor model for a tibial rat nerve. So the bounding of the electric field is more accurate. And here's a plot of, this is for a 16 micron diameter electrode, a fiber 8 micron diameter electrode, and you can see the amplitude increases as the separation decreases. And what's interesting, if we look at...
We have 1 minute left.
Okay, great. 4 different electrodes. The larger the diameter, the more spatial averaging you get near the electrode itself. So that's something that we might possibly worry about. However, again, if now you have a heterogeneous mixture, the fibers are kind of randomly situated with respect to the electrode. What happens in terms of how many number of nodes or number of fibers can we actually predict as a function of electrode diameter if we want our threshold voltage, which is going to be equal or to or above the signal. So for example, if our amplifier had a noise of 10 microvolts, and maybe we would want our signal noise ratio to be 2, so we would want to be able to record at least 20 microvolt signal. This is predicting that we would have about 4 fibers per electrode that could be recordable at that threshold level. And what's interesting is that in this case, it's not a function of fiber diameter or electrode diameter at all. You don't see that until you get to larger thresholds. And my last example, I'm working with Dr. Josh Wong and Aysegul Gunduz's students. She was doing the COMSOL modeling for us. Similar to what Cameron McIntyre is doing, we've been able to use pathway-specific fiber tracks and then predict the stimulation of those specific fiber tracks. And Dr. Wong wants to use this to determine the influence of different pulse waveforms used for DBS therapy for essential tremor patients. Can this modeling explain differences in therapy outcomes? And my last slide, I just want to say that physics-based modeling is very useful, but it's very computationally expensive. We could use numerical optimization techniques to really get optimal device designs or stimulus parameter designs, but we would need a much faster modeling platform. So could AI or deep learning be used to bypass these costly physics-based simulations? Be happy to hear your answers in the discussion. Thank you all.
Thank you so much, Dr. Patrick, for your talk. Now we invite the next speaker to share the screen. And let's try to keep our talk around 10 minutes so we can have enough time after all the talks to discuss and answer eventual question. So the next talk will be, will deep learning advance brain-human interface? Yes. So please, thank you.
Good morning, so many people virtually, and thank you for inviting me. I am particularly not a neuroengineering person, I'm a machine learning person. I started in machine learning long time back, and what I will be talking about is basically a quick review of what deep learning is and what could basically advance, how could deep learning advanced brain-machine interfaces. And you see in the title, deep learning is very small letters, because the way deep learning is being used for BMI, I believe that is just taking deep learning as it is and trying to apply. But advancement actually will require customized deep learning for a specific application, and I'll try to put what the emphasis could be. So with that, so this is the agenda, and first 4 items: brain-machine interface, machine learning, artificial neural networks and machine learning, and ANN and deep learning. Those are going to be just a basic introduction. I just included for completeness of the presentation, but I'll be quickly coming to the survey of application of deep learning to BMI, and then I will be actually presenting what I believe are the deepest contribution deep learning can make to BMI. So I think to this audience, what is BMI? I don't think I can explain, and I need to explain because you guys know better than what I do. So will this, the deep learning will bring it to a situation where people's intention will translate into the action or not? That is actually the most important thing. And I personally believe, yes, that will happen. It will go through many smaller steps and few big advances. And I believe deep learning has some potential to contribute to some bigger advantage. So what is machine learning? It's basically simple. You have a set of input examples and you want to define a function. That function basically takes the input and produce the desired output. In this instance, we are talking about input would be the brain signal. The intention in human mind will be sensed as a brain signal, and the function will learn how to activate action in the machine. So that is what in our case. So what is done in machine learning is basically to define the function, we define a long loss function, and closer the output to the intended output, the loss value is smaller. So just optimizing that loss function, we do machine learning. So if we look machine learning, we basically divide this into 2 different ways. Whether the learning is supervised, that means do we have association with input and output, or we don't. So that is the 2 horizontal lines you see. And then the other way is the input is continuous or discrete. So most of the success that we see and excitement is classification. That means supervised learning with discrete input. Now, if you go continuous input, then we do machine learning regression models. And then if we go unsupervised, so now we want to group the data in a way that each cluster forms one desired output. And then if data is continuous, instead of forming cluster, we try to find out area density. The higher density area, most likely one particular class that we want to learn. So with that, I'm not going to review different machine learning models. I will be directly giving a quick review of what is artificial neural networks and when it started. It started in 1958 in a publication in Nature, and Rosenberg actually what did create a model, very simple model is basically input to coming a summing unit pass through a threshold, and that can be a simple but effective computation model for a neuron. And he actually did show some applications of that model. But the problem of that model was that only one unit could define two boundary areas with one line. And how, if we have more than one label, how to actually learn, that was the problem. And that problem haunted people for many, many years. So in 1970, a MS thesis in Finland was published in Finnish, remained hidden. Then next, same thing, rediscovered backpropagation algorithm in a PhD dissertation in Harvard University Applied Math. And the committee thought that, okay, this gentleman has worked for 5 years, so it is a consolation PhD dissertation, it's okay. So that was basically 1974, and still it remained hidden until 1986, another Nature publication. These three gentlemen, what they did is not only rediscovered the back propagation algorithm, they did show some real applications and real good excitement started. And since then, only one machine learning algorithm that has been developed, which is the SVM. And if you try to see the application in biomedical engineering, in fact, almost any field, SVM is used blindly almost in everything. And backpropagation and neural network did not get any kind of highlight until 2012. So what is deep learning? To answer that, we have to basically look at the two steps of machine learning. One step is data engineering. So somehow pre-process the data, do the feature extraction, and then feed that to the machine learning model. You could do that for multiple models. So what deep learning really did is take the data engineering part, also put that into the part of the model. So deep learning is basically taking the data engineering part, which was basically handcrafted, was taken out and put together. First, it was in 1998 for actually US Postal Department used this model now for handwritten zip code detection, and it was developed in 1998, and it remained actually hidden. So what is actually different from backpropagation to this model is the feature engineering part. And if you look, the right side, 3 ribbons, that is what people used to use for backpropagation learning. And left side, you see that 4 volumetric area, those are basically for the feature extraction. So this is what we need to understand, where the real thing, real difference is. The architecture, the learning algorithm has not changed. So now, if we look at the feature extraction part, is it 2 layers or 4 layers? So the real answer is actually 2 layers, because we learn in 2, the other 2, there is no learning involved. It's just averaging or picking minimum or maximum. So last 2 layers are really the old machine learning part. So network was trained using backprocessing algorithm. Yes, feature extraction process was learned, not handcrafted like before. Unfortunately, this remained hidden. Excuse me. This remained hidden.
Sorry.
Go ahead.
You have less than 2 minutes, please.
Okay. So until 2012, this was not in public view. 2012 ImageNet competition, when actually this was discovered that, okay, it can do better than handcrafted and machine learning together, and this was the model. And since then, the deep learning has been in forefront of machine learning. So these are basically 3 areas where deep learning are used. The left blue side is the mainly where the deep learning now getting excitement. The green part, okay, but I believe that the impact that could be deep learning making for BMI is the yellow or orange portion there, generative models. And I'll be coming to that, why I think so. So generative models basically was discovered for giving network a better training, meaning that whatever learned, that sometime is memorized, and how to avoid that. So what is done is basically the real data is in the real image samples, and there is a data generator. You put some random input and it creates samples which is very similar than like real image. And this model discriminator try to classify whether it's real or fake. So eventually,
Dr. Sarkar, we have 1 minute. We want to make sure we stay on time.
I'm trying to. In fact, I don't have even 10 minutes if you are giving me 1 minute, but I will be finishing soon. Okay, so deep learning actually has been applied, and this is the latest survey that I found, and you see that application is really growing exponentially. And this up to 17, the data is complete, 20 and 19, data is incomplete. Out of all of those, most of the studies have used EZZ for the study. So only 2 publications used EZZ for data augmentation. This is one model. And what they did is they actually did try to augment data using the model that I was showing before. And with 100% data, this was the classification rate. And you see that using only 25% data, using the artificially generated data, this is how mass improvement happened. The other model that was 2017, that can we take people's intention and translate that into the images. And in this experiment, again, they use the data generator, and the data generator model actually created these images from people's thinking. Some of them were very successful, which is top 3 rows, and bottom 3 rows are not so good. Thank you very much. And these are the references.
All right, thank you very much for that talk, Dr. Sarkar. And we're going to keep moving right along. And next we'll be hearing about artistic brain-computer interfaces. So whenever you're able, go ahead and share your screen and just dive right in.
All right. Good morning, everyone. Thank you for inviting me. I'm Dr. Marvin Andujar. I'm a assistant professor in the computer science department and engineering at University of South Florida here in Tampa. So today I'll be talking to you about artistic brain-computer interfaces. And pretty much I'll provide a very quick background on what artistic BCIs are, and hoping to see, in case the participants are not familiar with it. And then I'll move along to talk a little bit about the enhanced application roadmap from the Brain-Computer Interface Society that they established a few years ago, and where we hope to be by the year 2025. Of course, that roadmap was created pre-COVID, so that might shift things a little bit. And then I'll provide 2 sample artistic BCI applications that we have been working in my research lab recently, and pretty much how they go with the enhanced application roadmap and where we hope to be by 2025. So very quickly, we pretty much know about these BCI cycle, right? So we know about it's a signal acquisition, feature extraction, feature translation, going through the signal processing pipeline, and then you send the control signals through machine learning for classification. And then Jonathan Walpole classified pretty much into 5 different applications. So that's what I meant by the enhanced application here. And as each a roadmap was created per application of the BCI Society. So as I mainly in my research lab, we concentrating from the enhanced application side. That's why I'll be talking more about that roadmap in a little bit. But very quickly, artistic BCI pretty much is a form of affective BCI. It's very interdisciplinary. It's looking to brain-computer interfaces and affective computing. And really, it's more about how can we look into brain activity and express it, having different forms of expression. It can be through audio in terms of music, brain-computer music interface, or use it as using visualization and as a stimulus, right? So even if we use media like videos and pictures and things like that, how can we stimulate people? So that's what we, and also from a passive BCI perspective, how can we understand better the affective and emotional state of users, and also even once we're able to post-process the data to be able to get an affective and emotional state from people, how can we represent that data in a manner that people can understand? When I mean people, I mean non-scientists, those who don't have that training. It can be visualizations that we are used to see in our mobile phones. When, like, if you have a Fitbit or a smartwatch, you're used to see that type of data in a daily basis. So these are some of the artistic BCI areas. Of course, not everything is in there that has identified that can actually contribute to the artistic BCI field. And many of these are also, of course, contribute for therapy use cases and also entertainment use cases, and also pretty much how can we improve our environment to improve the affective state of users. So, and here, this is where we go to the BCI roadmap for the enhanced application. In this use case that the society used, it was a student wearing pretty much sensors here on their head. Pretty much, in this case, we're going to just call them EEG sensors here. And while the student is doing some math here, you can see how their emotions start changing and how they start getting worried once they start doing certain problems that they don't feel too comfortable with. And that's what we would like to actually see more of. How can we start incorporating brain-computer interfaces, as we have seen more BCIs and they're becoming more wireless now? How can we, people start implementing these devices as wearable computers? Of course, the signal has to be robust and things like that, but this is the goal, where actual people outside the lab start using them. And in this case, how can students start using them as well? So if we look into this next roadmap, that I always say they just try to combine everything into just one picture. So I try to simplify as much as possible. The yellow here is pretty much academia, science and research, and our role is really to understand the user state and decode the brain state. Of course, somebody just talked about machine learning and utilizing and brain activity and signals and things like that. So that's where we should be. Then this purple here is the industry. So you can see here how we should be collaborating with industry, so they can help us improve the robustness of the signal acquisition, and the sensors, they should be improved as well. And then, of course, by 2025, we hope that we start at least testing how people start using like some sort of BCI apps, where we to actually get data about themselves, let's say even affective states, emotional states, or any other type of data besides control, right? So we referring all to passive BCI right now. We're not referring to anything about active BCI in terms of controlling machines like wheelchairs, or even a drone, because of my other work on brain control drones. So, but pretty much the goal here by 2025, in collaboration with industry, academia and industry, should be getting there, and through many different trials. So I'll be talking now that I gave you the roadmap where we want to be as a society by the year 2025. As pretty much, we try to shape our projects based on the vision of the BCI society. So in terms of what I call quantified self BCI, is any data that we can get from our body. Again, if you have a Fitbit smartwatch, if you already get heart rate from those devices, that's what we mean by quantified self. That's also known as personal informatics in computing. And I pretty much combine it with this called quantified self BCI, meaning any data we get from the brain is also considered quantified self, especially if we're getting useful information that can help people improve themselves. So in this picture here, the idea is that if somebody wearing a brain-computer interface and reading a piece of information, we're able to get data in real time and then post-process the data and send that data to a mobile phone, where we see the person see a visual feedback of their, let's say, the attention levels. And it also gives them intervention. The idea is like, okay, if somebody's not paying attention at a specific time, then they get a sign or a notification indicating, hey, you're not paying attention, you may want to take a break. It's good if you, because right now you're not really recording any information. And many of us, I think all of us have been, we have read the one page in a book, and then we look away. I really didn't pay any attention at all. I need to go back. So it would be nice to have some sort of notification like that. And of course, the state detection in this case is we use EEG caps. In this case, we are using Innovios or the GTEC devices. We may try to use wireless as we try to do testing in the lab and also outside the lab. And also is by post-processing these raw EEG data into the EEG waves and look into different frequencies to be able to understand the detect the affective states and evaluate them. And this is just more pretty much breaking down into the visualization and then the frequencies, which we may be familiar with, but I wanted to show you this so you can put things into perspective. And these are the engagement indices that we use, except for the last one, the FAS, which I'll talk a little bit about that later on. The top three, just pretty much ratios that we see in the literature that has been tested for engagement indices. So we try to evaluate all of them so we don't make any assumptions on which one works for each particular user for a particular task. And then for the second one, which is FAS, is a frontal alpha asymmetry. So we're looking in terms of motivation, how motivated the person is. So in this case here, we're trying to see if a person is motivated, plus also paying attention while they're trying to study or reading a book. Let's say if I need to study for a quiz tomorrow, I need to be as attentive, as motivated as I can to be able to grasp the information. And we're trying to get that information from the brain and be able to provide feedback to the user. So we provided some, we did a very quick study. Pretty much the purpose of the study was to see if there's if we show feedback to users obtained from the brain, is there will they try to self-regulate themselves the second time while they start doing the second learning task here. Will there be an improvement of attention? And something we realized is that with this engagement index, high beta over alpha, thank you. We noticed there's some increment of attention from the first task to the second task. Of course, now we're trying to do all these follow-up studies to make sure to see if that still stands, and to see if we find other engagement indices that we have not found before. Then moving to immersive brain painting is pretty much what I wanted to say here is this is also another artistic BCI application, and this is a more event-related potential P300 based project where they're creating abstract paintings in immersive environment, meaning in virtual reality environment, and they're using P300 as a stimulus where they concentrate on these symbols and they're able to create objects and be able to create change the colors, and all brain-based. And it has been very actually useful for those who with patients with ALS. So it allows them to express themselves, right? So once they have ALS, they're not able to paint, but able to brain paint. So they give them the ability to be able to express themselves creatively. So now we're looking to see how we can actually help those with ADHD here to see, because P300 tasks do require a lot of attention to do over time. So we're trying to see if ADHD, which is an NSF funded project, help them improve their attention retention over time. So based on event-related potential seems to be more useful than, let's say, motor imagery, at least based on the literature. And then lastly, what I want to show is pretty much this is the framework that we have used be able to come up with quick prototypes of, let's say, ERPs and motor imagery using a framework called OpenVibe, which is open source for brain-computer interface application. So we want to test things real quick. We pretty much use this type of framework. All right, thank you.
Thank you so much, Dr. Handuhar, for your talk. We're gonna move on with the next speaker. And please share the screen. And the talk is titled, Is Neural Microstimulation via Ultra Microelectrode Arrays Possible? So thank you. Go.
Great, thank you very much to the organizers. Can you hear me okay? Okay, great. 10 minutes is a tough slot. I'll do my best, but I really applaud you on putting together a great program. I've learned a lot. So my name is Kevin Otto. I am a professor at the University of Florida in the Biomedical Engineering Department, and I'm also the Senior Associate Chair. So the first thing I'd like to start off with, I want to express that our lab is committed to inclusion, diversity, equity, and access. And we're always recruiting, always looking for great people. I would appreciate following on Twitter if you want, but also contacting me directly via email. Okay, so microstimulation. Microstimulation has been an important understanding tool for science. It was first used by Wilder Penfield and later applied, well, I guess stimulation was first used by Wilder Penfield, but microstimulation was applied by Schmidt and Bach in this groundbreaking study in 1996, where they stimulated the visual cortex of human patients. In this case, one of the things that they saw was stimulation over the course of time actually resulted in decreased brightness. So while it was efficacious, it was interesting because it had this non-stationary component that needed to be understood, but it did provide a way to map out visual cortex and understand how phosphenes could be applied through microstimulation. So that's a good science example. How about prostheses? A lot of you are familiar, I'm sure, with this application out of the group at Pittsburgh, Rob Gaunt and Jen Collinger and others. This individual, Nathan, has 2 microelectrode arrays implanted into his brain. The first one is in his primary motor cortex that enables him to move this robot. The second is a stimulating array that's implanted into his somatosensory cortex. And what that allows him to do is sense touch from the robot. So in a second, when I play the movie and he has a fist bump from President Obama, he can actually feel that.
I don't know. I know the handshake that we got to the point where we can do fist bumps. Let's see what you got. Boom. Blow it up. You want to blow it up? All right, come on. That is unbelievable.
So I think it's clear that microstimulation has a future, both science and engineering-wise. What are potential circuits that might need microcircuit level manipulation? Depression is one that has had some success with DBS, but maybe more better treated with spatiotemporal microstimulation. Impulsivity and other neuropsychiatric, like obsessive compulsive disorder, social engagement disorder. Overeating in particular is another one that potentially could be treated through this. So why are these not on the market now? Why is it all macroelectrodes with DBS, and why are microelectrodes still in the research and clinical trial stage? They have a limited lifetime. And one of the reasons we think that is because they exhibit a reactive tissue response. At implantation, there is a glial cell recruitment, which encapsulates the electrode. That results in sometimes associated neuronal cell death and migration. From an engineering perspective, that looks like an impedance increase at the electrode, and ultimately an efficacy decrease. This has been pretty famously studied but in this paper by Polikoff in 2005. In this study, they had electrodes that were implanted here, and in these sections of tissue, you can see markers for microglia that have gathered around the electrode, astrocytes that are neighboring those microglia, and a lower neuron density shown by NUN and neurofilament shown here. My lab subsequently followed this up with an approach that we called device capture histology. And in this, we were able to capture the device in place in a 3-dimensional section. These were multi-labeled and so stained with 3 different markers. They were thick sections, approximately 400 microns thick, that were able to image through confocal microscopy. And what you see here is you see very similar things, but you definitely start to see a differential spatial arrangement. Near the surface of the brain, in this microglial stain, you can see a lot of microglia attached to the device, not as much in the deeper sites. This device, in this case, was only implanted for a week, so there hasn't been a huge astrocytic response developed to this yet. But you can start to see some astrocytic response, especially near the surface. And then one of the things that's very intriguing, but has not been studied that much, is the oligodendrocytic response. And essentially this demyelination that you see, which is depth penetrating all the way down to the deeper layers. So what does that mean? We see in previous studies, we've reported that stimulation performance can go down with time. So if we implant an electrode array into cortex in this longitudinal arrangement, so depth is spanned all the way down to the deepest layers of cortex, what we see is in longitudinal space, you can see the time axis is shown here, the depth axis is shown here, and the 2 takeaway points from this graph are that deeper is always better, And at any depth, time gets worse. So things get worse over time, but they get worse faster towards the surface. And in fact, this is a simple way to kind of group and quantify that. These are the deeper sites. These are the shallower sites. So the thresholds are going up. More recently in my group, we've actually extended this. We extended it a lot in terms of time, and the measurements are more precise because of a novel behavioral paradigm. And in this case, quantifying over 40 weeks, we see the layer 1, the uppermost sites, are failing at the pace that we expect. As you go deeper, it starts to get a little bit more stable until you get to layer 5. And at layer 5, it's actually pretty stable. It goes back up at layer 6. So what we know from other experiments is that microstimulation, or specifically microelectrode design, can influence that reactive tissue response, which we think could influence device lifetime for stimulation. Specifically, if we shift to ultra microelectrodes. And what does ultra mean? That means that the 2 of the 3 dimensions of a device are subcellular. They're sub 10 microns in dimension. So this was a paper by Seymour and Kipke in 2007, where they developed a device that had these little shanks out here. I like to call it an outrigger. It kind of, to me, looks like a catamaran boat as it goes in. And these little, this would be 4 microns by 5 microns on the edge out here. And when they did the histology, you can see that the histology surrounding these areas was actually much more normal compared to the thicker shank. This has been developed in many other instances, in this case by T.K. Kazai when he was with Daryl Kipke. He put in carbon electrodes that were smaller than 10 microns and in fact showed really remarkable longitudinal recording with these devices. Overall, these have been developed to develop arrays of microelectrodes. Here you have arrays of carbon fiber electrodes. Here you have novel substrates that are able to be in the ultra scale by Lan Luan and Changji. And then the NeuroPixels probe, in some ways, is an array of ultra microelectrodes. For those of you that don't know the NeuroPixels probe, this is a big consortium that developed a multi-channel microelectrode array. And technically, the electrodes are close to the ultra scale for the actual size of the devices. The depth of the device is close to the ultra scale. The width is not. The width is on the 100 micron scale. And then obviously in the 3rd dimension, it's not. So I'm not going to have any more answers for you necessarily, but I am going to ask the question, could we use high density ultra micro stimulation? We did a preliminary study to see if site size affected threshold. And so in this case, we designed an electrode array that had 4 different electrode site sizes on it, spanning from the smaller on microelectrode side up to a very large electrode. This is approximately the geometrical surface area of the Utah electrode array sites, the Black Rock arrays. And what we saw when you plot these relative to site size is we saw very similar thresholds over time. The thresholds do go down in the beginning, which is great. Maybe the animals are learning to do this experiment better. But after you get to 4 weeks, the performance is relatively stable. And in fact, if you break this same data out by depth, since we had 4 different depths, you do again see that depth effect where the most shallow sites actually have the worst performance and are the least stable. So one mechanism that we could potentially use for ultramicroelectrode stimulation is to use current steering, to use summated electrical currents from multiple channels at the same time, potentially in a spatial case like this, but also in potentially a temporal case where you're activating them faster and having the summation of the extracellular potential to allow excitation.
1 minute left.
Perfect. So I have one simple experiment that we did to show current steering in practice. This was not applied to an ultramicroelectrode case. This is simply 2 microelectrodes stimulated at the same time, and they're all plotted relative to a single microelectrode stimulated at a single time. So at the 0 case is 1 microelectrode stimulated at the same time, and we've normalized the amplitude of that electrode to 1. If you stimulate that site relative to its neighboring site, you see about a 47% reduction in amplitude to either site. I'll point out that the total charge here is actually more, but each individual site is seeing less. In this case, the total charge is actually a little bit less when you stimulate this site with this site. As you get further and further away, those fields sum less and less, and you start to see going back to having 2 individual electrodes stimulated at the same time. So this looks remarkably like a volume conductor in extracellular space. Basically, I think showing that we can use summated electrical stimulation from multiple channels to accomplish extracellular stimulation. So with that, I'd like to close thanking my lab, my collaborators, and definitely the funding source that enabled all of this work. And I look forward to questions in the open session.
Last but certainly not least on our list of speakers today will be telling us about closing the loop on essential tremor. So whenever you get the chance, please share your slides and take us home.
All right, so thank you for the invitation. It's been an excellent session. So I do have a disclosure. We actually received investigation device donations from Medtronic for this project. The topic of my presentation will be deep brain stimulation, and in fact, how do we create a bidirectional, BMI or bidirectional interface that could improve deep brain stimulation therapy. So once obscure, field some 30 years ago, now it's a very active scientific area of inquiry, not just because it actually has clinically proven to increase the quality of life for neurological disorders, but it also, the application of deep brain stimulation also allows us the opportunity for probing the function of circuits. But today within the clinic, the way the programming for deep brain stimulation or DBS is done is really doesn't have a scientific basis. So the clinicians basically observe the pathophysiology of the patient and they get their verbal feedback to be able to set these parameters, which is actually a large parameter space. So typically a 4-contact electrode is implanted in a deep brain structure thought to be involved in the pathological circuitry that causes the symptoms. And we call this conventional deep brain stimulation open loop because once in the clinic they set these parameters don't change until the patients come back for another clinical visit. So with that, our goal is to be able to measure these brain signals, basically find a marker that would allow us to maybe when to start the stimulation or how to change these parameters. And the devices that I said we received from Medtronic actually have basically chronic sensing capability from all the electrodes that are connected to it. So with that, this was basically a brain initiative project that we submitted to a program that was basically just in line with basically the work that was being done in my laboratory. So, but our idea here is to try to improve deep brain stimulation therapy for people with essential tremor. Essential tremor is the most common movement disorder, and DBS has been applied to it since the invention of deep brain stimulation. The technology really hasn't changed since those 30 years. There's actually a big subset of these patients that actually don't have tremor at rest. The tremor they have in their upper limbs basically start when they initiate a purposeful movement, and the sway is largest, the maximal at their actually intended target. So with that, our goal was to see if we could detect the intention of movement that would then basically tell the command for the electrode that's within the target that provides the symptom relief for these patients. And then once we don't detect the movement to the intention to move, the movement or tremor, we could basically then stop the stimulation. There's obviously many benefits to this. First of all, habituation in any kind of stimulation is a big problem. Our brains are really smart if they're being stimulated. With the same stimulus, it actually learns to ignore it. And then the other things is deep brain stimulation does cause a lot of side effects in these patients. So if we can deliver the stimulation only when necessary, we hope that we can alleviate these problems and of course elongate battery life. Okay, with that in mind, so the human motor system actually has been studied really well with epilepsy patients. So these patients typically go through two-stage brain surgery and they receive a lot of subdural electrodes over large areas of their brain. So we in fact know since the late 90s that within our motor system, within our primary motor cortex and our premotor cortex, we basically see these low frequency band desynchronizations when someone executes a movement or it plans on making this movement. We have these high frequency band synchronizations, and these high frequency bands actually are very localized. They basically co-localize with electrical stimulation of the area, whereas a low frequency band desynchronizations actually happen in a much broader area, and this is probably the reason why we see these sensory-motor rhythm changes in non-invasive EEG as well. So with this knowledge, we propose to place a much smaller subdural cortical strip over the premotor and motor cortex within these patients. So again, this will give us that robust signal that will tell us when to start stimulation and end the stimulation. But the other advantage that it brings is that it is far away from the stimulation area, so we are actually won't be receiving much of the stimulation artifacts that we do in fact see on the depth signal. Okay, so with that, so this is some data from one of our earliest patients. So the targeted area for essential tremor DBS therapy is the motor nucleus of the thalamus, the ventral intermediate nucleus. And then, like I said, we basically targeted the premotor and primary motor areas for the cortical probe. So at the very top and bottom, we have some contralateral and ipsilateral hand acceleration. So this laterality is basically based on where the implants are. So when we say contralateral, this is contralateral to the implant. And so these are the time domain signals, and we created a spectrogram of these. And both with ipsilateral and contralateral hand activity, we basically see these low frequency band desynchronizations, both in the cortex and actually the thalamic signal itself. This is really intriguing, but with these devices, once the stimulation is on, we actually don't see this signal anymore. And we do see the high frequency band synchronization on the cortical with only the contralateral hand movement. But again, with these devices, they're first generation, so their noise floor isn't too reliable. So actually the low frequency band signal is more reliable. And we basically also found that actually whether it's ipsi or contralateral hand, these people typically have tremor on both hands. So that basically won't be a disadvantage of triggering stimulation with the ipsilateral hand activity. So with that, we basically tested, we now have completed the study with 3 participants. So this is actually a left-handed gentleman with essential tremor. Here is the black boxes are basically the wireless sensors that they're wearing. And so because they're left-hand dominant, their implants were on their right hemisphere. And so you can basically see that they are suffering from tremor. Just to save some water, I'll pass this slide. And don't worry that you're not hearing anything with the videos. I intentionally removed the audio so we wouldn't have any echoing problems. So before I start this video, let me tell you what you're going to be seeing on the right-hand side. So this basically block is the raw channel. This is again the cortical signal. And then this block, the one below it, is basically this signal separated into two beta bands. So a low beta band that's centered around 15 hertz with a 5 hertz bandwidth, and this is one with a center of 25 hertz, again with a 5 hertz bandwidth. So together they have to desynchronize basically to give us the detection of a movement intention. So once that is detected, this panel turns lilac, I guess. And then here we have the right hand EMG and the left hand EMG. Again, the left hand is the contralateral to their implant, and again, their dominant hand. And basically this stimulation, I guess, subfigure basically rises up whenever there's a detection, and it basically tells us that, in fact, stimulation is being delivered. And this is all in sync with the video of the subject. So here the movement was detected, and you can see, or the intention was detected, and you can see that they were in fact able to basically transfer this water from one cup to the other. Again, we see the co-desync of these bands, and we kind of see that actually, slightly happens even before the EMG activity. And again, here, this was a good example that it happened before the EMG activity. And basically it sustains, the stimulation sustains throughout the movement. And once they went back to rest, both bands again go back to their initial level, they rise, and so on and so forth. So like I said, we've completed this study in 3 subjects right now, and these are their clinical scores. So TRS is the tremor rating scale, and the lower it is, it means that the severity of the tremor is less. So our goal is to be able to decrease this as much as we can. And here we basically have been working with them over 6 months after their surgery. They come back to our lab once a month for 2 days. And here are their tremor scales when the DBS is turned off. So you can see that even an open loop DBS, obviously as expected, reduces their scores.
1 minute, thank you.
Yes, and their closed loop basically scores are just as comparable. So we're basically saying they're just as efficacious. And if we look at their energy savings, we're about, on average, able to, save 50% of the battery usage. So with that, I do want to thank the funding source. So this is funded by the BRAIN Initiative. Again, like I said, Medtronic provides the device technicians for investigational use only. And I really have to thank my collaborators. Mike Okun is from the Department of Neurology, and Kelly Foot is a neurosurgeon. Together, they co-direct the Fixed Cell Center for Neurological Disorders at UF. I do want to thank all my laboratory members, especially Erin who has received his PhD and who's now doing the postdoctoral fellowship. He's the first author on a paper that is accepted finally to this Science Translational Medicine. So we're hoping that it's going to be published in the December issues. And with that, I'm happy to take the questions in the Q&A session.
Thank you so much. Thank you for all the beautiful talks that all the panelists have given. Sadly, we are a little over time. So if anyone has questions, please ask them now in the Q&A box.